Welcome friends to a new session on extensions to Mendelian genetics. In this session, we will discuss about the multiple gene inheritance. First, let us see about quantitative inheritance. We have studied the inheritance of some Mendelian character earlier. In that studies, we can easily classify distinct characters such as purple and white flower color, green pea pod and yellow pea pod and many more similar examples, which are collectively known as discontinuous variation. However, some characteristics such as skin color, eye color and height in humans in a population appears continuously, generally show a continuous variation. These characters are defined as quantitative when they appear to be continuous, so that individual function of genes in the final phenotype is not known. These features are controlled by many genes and therefore categorized as quantitative inheritance or polygenic inheritance. In this type of inheritance, more than one pair of genes controls a single phenotypic trait. This phenotypic trait is called multi-factor or polygenic trait, and the inheritance pattern is called quantitative or polygenic inheritance. In this, the F1 individuals are usually intermediate between the two parents and are very much similar to one another. Sir Francis Galton, who is a fellow of Royal Society, a protogenetist, psychometrician and a statistician, was the first to apply statistical methods to the study of human differences and inheritance of intelligence. And Carl Pearson, again a fellow of Royal Society, known for Pearson's chi-square test, studied some human traits such as intelligent quotient, height and weight by some statistical techniques in the 19th century. In case of continuous variation, the number of genotypes increases with increase in the number of alleles at each locus. So, each genotype in quantitative inheritance gives rise to a separate phenotypic appearance with slight difference. For instance, a cross between two F1 individuals results a highly variable F2 progeny, in which a few individuals resemble one grandparent, a few resemble the other grandparent and the remaining ranging between the two. The assumptions that can be made for understanding the quantitative inheritance are: 1. A series of contributory and non-contributory alleles are involved in a cumulative way. 2. Effect of each contributory allele is additive. 3. No genetic interaction of alleles take place between alleles of different loci. 4. No linkage is observed between loci involved. 5. Non-allelic pair exhibit dominance. 6. Considerable effect is due to environmental factors in the phenotypic expression of multifactorial genes. These assumptions are reflected in the quantitative inheritance we are going to study henceforth. The quantitative inheritance will be explained in this section with the examples of skin color in human and ear size in corn. First, let us see about skin color in human. The inheritance of skin color in human beings is the classical example of quantitative inheritance. The visual appearance of human skin is determined by four major pigments: the oxygenated, which is red color, and reduced hemoglobins of the dermal blood vessels, which is blue color, the carotenoids, yellow color, and melanin. Melanin is found primarily in the epidermis. If melanin is present in sufficient concentration, it can hide the hemoglobin pigments and then vessels are under the layer of epidermis. Human geneticists used skin color as an example of human quantitative trait because the basic biology of melanin production and the method of measurement are well advanced. Melanin is the one of the most important substance responsible for skin pigmentation. It is the main pigment of the skin color of darker skinned humans, which is produced within the skin in cells called melanocytes. Human skin color ranges in variety from the darkest brown to the lightest pinkish white. The light skin in humans is determined mainly by bluish white connective tissue under the dermis and by hemoglobin circulating in the veins of the dermis. Sometimes, especially on the face, the red color in the skin becomes very visible. As a consequence of physical exercise or the stimulation of the nerve system due to anger, fear, as well as the arterioles when dilates. The neural origin of the melanocytes is reflected in the retention of dendritic processes, which are inserted between neighboring epidermal cells called keratinocytes or Malphigian cells. Keratinocytes produce a fibrous protein called keratin. Keratin also form the outer dead layer of the skin called the stratum corneum as they flatten and die. Melanocyte is a one-celled secretory gland supplying melanin to approximately 36 keratinocytes. Melanin is produced within specialized melanocytes organelles called melanosomes. Melanosomes or pre-melanosomes are formed and filled with a fibrous protein matrix. Then a chemical reaction catalyzed by the enzyme tyrosinase causes oxidation of amino acid tyrosine, which then becomes 3,4-dihydroxyphenylalanine. In short, we call it as DOPA. Tyrosinase also catalyzes the conversion of DOPA to dopaquinone and results in the formation of melanin. Finally, melanin is deposited onto the fibrous matrix in the melanosome. Once the protein matrix is filled with the melanin, the melanosomes migrate to dendritic processes of the melanocyte. From there, an adjacent keratinocyte engulfs the dendrite. The dendritic wall disappears and the melanin granules enter the keratinocyte cytoplasm, either singly or in clumps of 2 or more. In Caucasians, the granules tend to be small and are aggregated into lysosome-like organelles. With these organelles, the granules often become fragmented and degraded. In blacks, melanin granules tend to be larger and reach the keratinocytes as single granules that apparently do not decompose. The process of transfer from melanocyte to keratinocyte is under the control of both cells. Although exposure to ultraviolet light stimulates production and incorporation of melanin in vivo, melanocytes isolated in vitro do not produce melanin in response to UV light. In early studies, the skin color were measured by crude measurement techniques. In some studies, the skin color was measured by matching the colored paper standards and with the tinted enamel and the pupil's color top. Originally designed as a toy which allowed more continuous measurement. The first step of the biosynthetic pathway for both eumelanins and pheomelanins is catalyzed by tyrosinase. Tyrosine give rise to DOPA and DOPA finally to dopaquinone. Dopaquinone can combine with cysteine by 2 pathways to benzathiazines and pheomelanins. Dopaquinone combines with cysteine to form 5,6-cysteanyl DOPA, which give rise to benzothiazine intermediate and finally to pheomelanin. Similarly, dopaquinone combines with cysteine to form 2,6-cystenyl dopa, which give rise to benzothiazine intermediate, and finally to pheomelanin. Also, dopaquinone can be converted to leukodopachrome and follow two more pathways to the eumelanins. Dopaquinone give rise to leukodopachrome, which gets converted to dopachrome, which later converts to 5,6-dihydroxyindole 2-carboxylic acid, which later get converted to quinone and finally to eumelanin. In another pathway, dopaquinone gets converted into leukodopachrome, which gets converted into dopachrome, which later converts into 5,6-dihydroxyindole, and it is converted into quinone and finally to eumelanin. The skin color in human is under the control of at least 3 independently inheriting genes. The genes with the dominant alleles A, B and C contribute 1 unit of dark shade in the phenotype, and each will be completely dominant over its respective recessive allele small a, small b and small c. Capital A and small a, capital B small b and capital C small c are located in different chromosomes. The genes capital A, B and C, which controls dark skin color, are incompletely dominant. The darkness of human skin color is proportional to the presence of total number of the dominant genes. All the 6 dominant genes capital A, capital B and capital C are present in a very dark human. A human with very light skin color has all the 6 recessive genes small a, small b, small c. An offspring with capital A, small a, capital B, small b, capital C, small c, or capital A, capital B, small b and small c genotypes have 3 units for skin shades. The skin color in humans was studied by Charles Benedict Davenport, a prominent supporter of Mendelian inheritance. He was one of the leaders of the American eugenics movement and prominent American eugenicist and biologist. He assumed that the dominant gene contributes a fixed amount of melanin and the amount is proportional to the number of dominant genes. He suggested the presence of 2 pairs of genes capital A small a and capital B small b, so that the dark skin have the genotype capital A, capital B. White skin have all A and B as small, and the offspring of the 2 dominant gene with a genotype capital A, capital B, small b have an intermediate color called mulatto. In the table and figure, it shows crossing of 2 heterozygous capital A, capital B, small b, capital C, small c parents having 3 dominant genes and 3 recessive genes. Of these crosses, 64 progenies are formed. The phenotypes represent 7 different types. The 7 shades of darkness of skin is represented over here. To the extreme left, it is totally light shade, and to the middle it is the mix of both extreme colors represented by 20 numbers, and towards the extreme right is the dark color, which is represented just by 1 number. If this data is put together as graph, it will represent as a bell shaped curve. Ear size in corn. Maize, otherwise known as corn, is a large grain plant. The leafy stalk produces ears which contain the grain, which are seeds called kernels. The ear shoot was developed from an axillary meristem, which are initiated very early at multiple stalk nodes in the growth of a corn plant. Row number and kernel number per row are the two important factors in ear size determination in corn. In a corn plant, typically 750 to 1000 ovules develop on each ear shoot. Rollins Adams Emerson, an American genetist who rediscovered the laws of inheritance established by Gregor Mendel and Edward Murray East, studied ear length in corn in the year 1913. The ear length of corn shows quantitative inheritance. They crossed two pure breeding strains of corn with little variation in ear length. The variations are: 1. Tom Thumb popcorn has long ears with a mean length 16.8 centimeter and a standard deviation 1.887. Second, the black Mexican sweet corn variety has short ears with a mean length 6.63 centimeter and a standard deviation of 0.816. The two strains were crossed and the F1 plants are interbred. The mean ear length in the F1 plant is 12.12 centimeter, approximately intermediate, and standard deviation is 1.519. Since the parents were true breeding, all the plants have the same heterozygous genotype in F1 generation, and any variation in length is due to environmental factors. The F1 plants were crossed. The mean ear length of the F2 plant is 12.89 centimeter, very similar to the F1 plants, but in the F2 plant the standard deviation is 2.252 centimeter, reflecting its greater variability. From the experiment, it is expected that the variability in ear length from plant to plant is due to environmental factors in addition to quantitative inheritance. However, to understand the mechanism of quantitative inheritance, we ignore the effect of environment. The increased variability in the F2 plants results from its greater genetic variation. We usually find the mean value of quantitative inheritance in the F1 plants between the mean of the true breeding parents. Identification of the exact number of genes involved in quantitative or polygenic inheritance has not been exactly established in many cases. The multiple gene hypothesis suggests that a number of factors contribute the complex form of inheritance. However, it is confirmed that the quantitative inheritance is influenced by many genes. Each of the genes contribute small but additive effect. We explained the examples on the basis of action and segregation of alleles at a number of loci having small and additive effects. Now, let us see something about transgressive variation. Transgressive segregation is the formation of extreme phenotypes or transgressive phenotypes observed in segregated hybrid populations compared to phenotypes observed in the parental lines. In other words, transgressive variation is the production of offspring with the phenotypic characteristics such as number of seeds per plant or flowering time that exceed those of the parents, or it is the formation of transgressive phenotypes or extreme phenotypes. Transgressive segregation can allow a hybrid species to populate different environments, niches in which the parent species do not reside or compete in the existing environment with the parental species. This generally results from the interaction between genes present in the two different parents. For instance, genes from any one of the parent act as master switches or activators at the regulatory network that leads to a phenotype. These phenotypes fall outside the range of the parents. A dark red represented by all capital R and white represented by all small r, grain color in wheat were crossed, and all the progenies produced in the F1 generation have the phenotypic character medium red, and the genetic constitution is capital R, small r, capital R, small r. But in the F2 generation, all the progenies show a variation in the phenotypic expression. In F2 generation, the grain color in wheat results in the genotypic ratio 1:4:6:4:1, and the phenotypes are dark red represented by all capital R, medium dark red represented by 3 capital R and 1 small r, medium red represented by 2 capital R and 2 small r, and light red represented by 1 capital R and 3 small r, and white represented completely by small r. Similar observations are made in quantitative traits such as yield. It is often observed in the progenies that are derived from interspecific transgression. There are 3 explanations for the interspecific transgression. 1. Induced de novo mutation in the white cross itself. 2. Complementary action of the genes from the 2 parental species. And 3. Unmasking of recessive genes in the heterozygous in the wild species. Transgressive variation might be a key to the improvement of cultivated plants having extreme phenotypes. It is the generation of F2 or hybrid progeny with the phenotypes that fall outside the phenotypic characteristic of parental populations from which they are derived. The improvement of self-pollinated crops, example barley, is connected with the production of homozygous populations superior to the genotypes of the parents. Transgressive individuals observed in early generations, that is F2 and F3, may be heterozygous. It is widely known that the frequency of the effect of transgression in homozygous population depends on the genotypes of the crossing parents. Transgressive variation might be an important mechanism prompting sudden phenotypic changes and ecological transitions in evolution. If one could predict and manipulate this type of genetic variation, it might be harnessed to produce crop varieties with novel and exceptional characteristics. Moreover, the transgressive individuals may possess characteristics that will allow them to occupy a particular ecological niche. In plant hybrids, transgressive variation appears to be ubiquitous. In plants, a remarkable diversity of traits has been shown to exhibit transgressive variation. Various biotic and abiotic factors influence the diversity of morphological traits. Transgressive variation does occur in animals, but it occurs to be much less frequent than in plants. Transgressive variation may be a major source of novel adaptations in hybrid. The major explanations for transgressive phenotypes in segregating hybrid populations include: 1. Reduced developmental stability, 2. Advanced rate of mutation in hybrids, 3. The unmasking of rare recessive alleles that are normally heterozygous in the parental taxa, 4. Overdominance or non-additivity of allelic effects within a locus, 5. Epistasis or non-additivity of allelic effects between loci, 6. The complementary action of additive alleles that are dispersed between the parental lines, and 7. variation in the chromosome number. Quantitative trait loci, which we call QTL, and Dunn's test are the important and most common test for the determination of transgressive segregation occurred within a population. The Dunn's test reveals that the performance of hybrid species was different from the parent species by looking whether or not the mean of the parent species significantly differs from mean of the other groups. If any difference is there, that is an indication of transgressive segregation. The relevance of Dunn's test is that it is a special case of multiple comparisons where all factor level means are compared to the control, but no other pairs of means are tested. A QTL is defined as a region of the genome that is associated with an effect on a quantitative trait. The relevance of QTL is that it is widely used in crop plants to analyze for diverse traits including yield, quality, disease and insect resistance, abiotic stress tolerance and environmental adaptation. Alleles with quantitative trait loci that were opposed either by underdominance or overdominance of the parental QTL indicate that transgressive segregation occurred. But alleles with QTL that was the same as the predicted parental QTL show that there was no transgressive segregation. It creates a new hybrid species or novel species to arise that are more fit than their parents. Transgressive segregation is used in genetic engineering for the creation of new organism that is more fit than the last. Now, let us summarize the discussion over here on quantitative inheritance. Quantitative inheritance is concerned with characters controlled by many genes called quantitative genes or polygenes. Inheritance of these genes is completely dominant, so that the heterozygote is an intermediate between the parent's phenotype. Normally, dominant and recessive allele or alleles of the genes might be expressed, but the dominant allele would be active and the recessive one inactive. Many examples of skin, eye color and height in humans and other examples from plants and animals show phenotypes with a continuous variation. Such features are usually controlled by two or more genes and hence called polygenic inheritance or quantitative inheritance. A very good example of quantitative inheritance is skin color in human. Skin color in human is determined by three alleles A, B and C, which tend to be extremely dark when all these are in capital, and when all these are in small letters they are extremely light. In between there are continuous variations of skin color such as very dark, dark, intermediate, light and very light. Each allele is providing one dose of pigment. Similar analogy is extended to many examples. Another example for polygenic inheritance is ear size in corn. In addition to quantitative inheritance, the variability in ear length from plant to plant is due to environmental factors. The production of offspring with the phenotypic traits that exceed those of the parents is known as transgressive variation. Quantitative trait loci QTL and Dunn's test is used to measure the occurrence of continuous variation of a trait in a population controlled by several pairs of genes. Now, I will give you some assignments to work out. 1. Give a detailed account of polygenic inheritance with suitable examples. 2. Explain inheritance of skin pigmentation in humans. 3. Write notes on ear length in corn. 4. What is the genetic basis of quantitative traits? 5. What is the difference between Mendelian traits and quantitative traits? Now, let us see some references which you can refer to for the part which we have already discussed. The first is a research paper entitled Role of Light in Human Skin Color Variation, authored by Quevedo, Fitzpatrick, Pathak, Jimbo in the year 1975, published in American Journal of Physiology Anthropology. The volume number is 43, page numbers are from 393 to 408. Another publication is entitled Effect of Environment on the Early Steps of Ear Initiation in Maize, authored by Lugin and Bernier in the year 1996, and which has come up in Plant Cell and Environment. It is a very prominent journal. The volume number is 19, page numbers are 217 to 224. Next is entitled Inflorescence of Maize, Wheat, Rye, Barley and Oats, Their Initiation and Development, authored by Bonnet in the year 1966, and which has come up in Bulletin 721, University of Illinois College of Agriculture, Agricultural Experimental Station. A fourth reference would be a book entitled Principles of Genetics, authored by Gardner, Simmons, Nustad in the year 2008. It is the 8th edition, published by Ville, India. A fifth reference would be Principles of Genetics, 5th edition, authored by Nustad, Simmons in the year 2009, published by John Ville and Sons. Sixth reference is entitled Concepts of Genetics, 10th edition, authored by Clark, Cummings and Spencer in the year 2012 and published by Benjamin Cummings. A seventh reference is entitled Genetics, a Molecular Approach, 3rd edition. The editors are Benjamin Cummings, Griffiths, Wessler, Levontin, Carroll. The author of this publication is Russell. It is published by Freeman and Company. The websites where you can find more information on this is www.genetics.org. Another website is www.genome.gov. Thank you for watching this session. See you next time. Until then, bye.